Welcome to AP Biology. Today I want to go over the 3 steps of cell signaling. The first is reception, or signal perception, and then transduction, which is a series of changes that take place between reception and response. And then the response is finally what happens at the end. And so here's a visual of that. Here's a signaling molecule. We'll also call that a ligand or a ligand. That's got a very special shape, and it fits in with one particular receptor type. And so this is just showing you the shape of some signaling molecule and that it fits into its particular receptor. So if this molecule is insulin, this receptor would be just for insulin. This receptor wouldn't fit with serotonin or acetylcholine or epinephrine or anything else. It would just fit with its one type of signaling molecule. So that is reception or signal perception. If a cell doesn't have the receptor for the signaling molecule, then nothing happens. The signaling molecule can't affect it. And then we have signal transduction. Transduction means that the signal is changed. So it's changed from this molecule to a series of chemical reactions that happen. So the first thing that happens when the ligand binds to the receptor is that the receptor typically will change shape inside. Usually it becomes activated, and that change in shape, maybe it's an enzyme now, that change in shape can activate another enzyme or some other molecule, which activates another, which activates another. So these arrows mean it proceeds forward or the next thing is activated. Sometimes we'll call these relay molecules. It's sort of like a relay race where you hand off a baton. The first guy has to do something, and then the next one, and then the next one. And then finally we get some kind of cellular response. So the response might be that the cardiac muscle cells have to pump harder, or that the smooth muscles in your eyes relax, or that glycogen is broken down to glucose, or that glucose is taken into a cell, or that mitosis happens. There can be a whole bunch of cellular responses. So what we're going to do in this unit is look at 4 different types of receptors, and then we'll look at examples of transduction pathways. And there'll be some vocab specific to each of them, and then we'll look at some examples of responses. So let's start with reception. A signaling molecule, or a ligand, binds to a receptor protein, causing it to change shape. So here's the receptor, and again, they're very highly specific. The ligand will fit right here into the receptor, and once they bind, typically it's going to cause a shape change here. So just like when we did the enzyme unit, do you remember how if there's an enzyme and its active site isn't quite right for the substrate, if there's another molecule that binds here, maybe it's an activator, and it changes the shape of the active site, so now the enzyme can bind with the substrate. So that's kind of what's happening here. The ligand binds to the receptor, and it can activate or can change the shape of the inside of the molecule here, the inside of the receptor. So the ligand binding between the chemical messenger, or the binding between the chemical messenger, the ligand, and the receptor is highly specific. It's a one-to-one relationship. So if this is insulin, this receptor is for only insulin. The ligand can be a peptide, some other small chemical, or a protein. So remember, a protein is made out of amino acids bonded together and then folded into a 3-dimensional structure. A peptide is like that, but it's smaller, so it might be 3 or 4 or 5 or maybe even 10 amino acids, but it's not a big protein. Or it could be some other small chemical, like maybe it's estrogen, which would have the general steroid shape, which is just a hydrocarbon, a bunch of carbons attached to hydrogens. So it would look something like this, and I think some of them have like an OH here or there, some carbons over here. So that could be a type of ligand, or it could be, I don't know, I think some of them just have like a, just one ring and then maybe a couple of carbons or something. So that could be the shape of another ligand molecule. So there are a bunch of different types. So then what happens is you get a change of shape in here. Usually it's activated. You might see the term conformational change. Your conformation is just your shape. So these 2 things mean the same thing. It's usually the initial transduction of the signal, meaning the ligand first has to bind to the receptor, but the transduction are the changes that happen after. The first change is typically the receptor changing shape. Most signal receptors are plasma membrane proteins. They're usually on the surface here. Some of them, some receptors are actually in the inside, and that would be a receptor that's on the inside would be anything that can bind to a ligand if the ligand can go right through. So there are a couple of different major types of ligands. Some of them are water soluble or hydrophilic, and some of them are not. They're lipid soluble, they're hydrophobic. So you might remember that the lipid bilayer, the cell membrane is made out of a lipid bilayer mainly, with some embedded proteins and carbs. And the lipid bilayer looks like this. Remember, it's got those phospholipids, that's one layer, and then it has another layer, right, like this. And so these parts, the faces there, they're all smiling. They're all saying, oh my God, I love water. Because outside the cell, there's water, and inside the cell in the cytoplasm there's water, right? So there's water out here and water out here. But this part is a lipid. This part is hydrophobic. And so hydrophobic molecules can go right through the lipid bilayer. So molecules like testosterone, those are steroids, and they mix right with this hydrophobic membrane and can go right through. So the receptor for testosterone would not be on the surface like it is here. The receptor for the testosterone, for the hydrophobic ligands, the receptors are going to be inside. They're either inside the cell or even further inside the cell in the nucleus. So receptors are inside the cell. Whereas a hydrophilic, a water-soluble ligand would be it wouldn't be going through the lipid bilayer, right? The lipids would just reject it, not allow it to go through. So the receptors are typically going to be found on the surface. So receptors, they're on the surface of the cell. So we're going to talk about 3 different major categories of receptors that are on the surface, and then we'll talk briefly about the ones that are inside.